Rick is a project scientist in the Ecology Department at the Lawrence Berkeley National Lab. See, we get wicked smart people here at NCRA. And he's going to speak to you about the fate of pharmaceuticals in thermophilic compost. I just skipped over your whole bio, but it'll be great.
That's fine. Well, thank you very much. I'm a first timer here. Normally I go to conferences with much less interesting people who talk about science, and you guys are on the ground doing fantastic things, and it's wonderful to be here, so thanks for having me. So I work up at the Lawrence Berkeley lab, just up the hill from here, behind campus, and this is the beautiful view I would have if I wasn't housed in a concrete bunker with no windows, but you know, while I may not be the most interesting man in the world, I work on the most interesting organisms in the world, which are microbes. And I want to talk a little bit about why we're so fascinated by these bacteria and fungi. And you know, one problem we think that they can help us with, and we want to understand the extent to which they can do this, is to address this problem that we've been discussing in the last few talks, which is widespread pharmaceutical waste. And as you've heard, it's a major problem. And just one more alarming statistic to add to what you've already heard is that, you know, we're finding pharmaceuticals just about everywhere in the environment. 80% of US streams have detectable levels of pharmaceuticals. And clearly we need to address this problem in a wide variety of ways, both in terms of improving our disposal methods, treating waste that contain pharmaceuticals, and we're just never going to get it completely out of the waste stream because we're all going to just keep taking drugs and it's going to end up in the toilet and it's going to get flushed down the drain. And you know, we need to really, this is an emerging problem, and as we try to, you know, reuse our waste for beneficial uses, the question is going to come up: how well have you removed pharmaceuticals and addressed this problem? And so what we're focused on is, can the bugs eat our drugs? And we know bugs can do a lot of things. I mean, this is the beauty of compost. It's a microbial process, and we've got a whole bunch of voracious microorganisms in compost eating away. And we want to know, when you mix in and you've got compost that contain pharmaceuticals, how well do the microorganisms consume different classes of pharmaceuticals? And so we've been really gearing up our laboratory up the hill to do this research. Just a little bit about my background, I'm a microbial ecologist. I tend to work on soils, interested in microorganisms from the perspective of, you have these complex microbial systems with a variety of organisms. How do they affect carbon cycling? How do they affect nutrient cycling? And how can they help us with bioremediation? And these are we've been addressing, you know, global scale problems of the carbon cycle. And, you know, we've really started drilling down on this problem because we realized that, you know, part of the solution to the carbon crisis that we have on the planet is to reuse our wastes. And so we think we can kind of take some of these nifty tools that we've developed up at the National Lab and start applying them to this problem on the ground. And I want to talk about how we've been starting work with some agencies and municipalities to do this. So last year we received some funding from Stop Waste, as well as the San Francisco Department of the Environment, to do some preliminary experiments looking at pharmaceutical degradation in compost piles. So we're talking about thermophilic compost. The heat generated from the piles is all due to the microbial activity. And so there's a heat component, there's a microbial enzyme component to the degradation, and we want to really understand how it is mechanistically that the microbes are degrading different types of pharmaceuticals and how effectively they're doing it. And so the microbes they didn't evolve growing up on psychoactive drugs. Typically, most microbes haven't seen this in the course of their daily lives. But the way I think about it is they do this co-metabolically, kind of fortuitously, where you can think of, like, if I gave you a bowl of really yummy tomato soup, and you're hungry, you're going to just chow it down. And let's just say this is sawdust right here. It's actually Parmesan cheese, I think, but just use your imagination. So if I gave you that on a plate, you wouldn't want to eat it. But if I mixed it into the soup and I didn't really tell you about it, you'd consume it, and you might, degrade it. And so really, it's, if in a compost pile, the microbes are voraciously consuming what they've been given in terms of the plant products and carbohydrates and so forth. But in the course of producing the enzymes to degrade those things, they can also, those enzymes can be used to degrade pharmaceuticals in certain cases. And so we want to understand this better. And so in conjunction with Musahid Ahmed, who's a chemist who works in the Chemistry Sciences Division at Lawrence Berkeley Lab, he's really outfitted his laboratory with this new instrumentation to do very sensitive detection of pharmaceuticals in complex environmental samples. And part of the reason that we don't know a lot about what happens to the fate of pharmaceuticals in the environment is because they have been really hard to measure. It's really hard to detect low levels of pharmaceuticals when you have a whole host of other interfering compounds. And so really the technology has been coming online in the last decade to be able to do these measurements. And very little has been done in relationship to compost. And so it's really, from a scientific perspective, very new. And then I think from your perspective, it's useful information to really address what happens when you have pharmaceuticals in composting. Can composting actually be used as a potential alternative to incineration or other disposal methods for some classes of pharmaceuticals? So what we've set up is some experimental piles. This is an experimental setup we have up in Marin County in conjunction with the Rathman Foundation, who's been doing a lot of interesting research on compost. And we've been basically spiking the piles with pharmaceuticals and looking at the fate of those pharmaceuticals over time. And now we're doing this up at our lab, which is a lot more convenient, but a lot less picturesque. And so we're generating data like this, where we're looking at the rate of disappearance and consumption of the pharmaceutical. So here's an example. This is ibuprofen. But we also want to know what happens to the original compound once it disappears. What are the daughter products that are being generated? And are those safe? Are we comfortable with those in our waste? What is the ultimate fate of the compound as it's being degraded in the compost pile? And then the work that I do is really focused in on the microorganisms themselves and identifying who they are and mechanistically how they carry out the degradation process. And so this is just a graph showing in one of our piles the sequence of organisms that come up over the course of a 7 or 8 week experiment. And there's a whole symphony of microbial players, as you know, that happen in thermophilic compost. And so we've developed methods in our laboratory to use molecular biology to identify every one of those microorganisms simultaneously. And then we can even dig deeper into their genomes to understand what their metabolic capabilities are, and to put that together with the chemical information to really develop an understanding of how the process works so that we can then begin to optimize it. And so right now we're really just in sort of this learning phase, this methods development phase. But, and I just want to also mention that, in conjunction with this, we look at the destruction of pathogens. So we also want to make sure that the compost is safe biologically. And so again, these tools are really powerful because we look at everything simultaneously. And we're also working towards the future, which is taking all this sophisticated instrumentation that we have up in the laboratory and reducing it down to something that we can take out into the field. And that hopefully, ultimately, we can put in the hands of people that aren't experts. And so what's exciting for us, and which really traditionally hasn't been done at the laboratory, is working with municipalities and agencies. You know, typically up at the lab, we get these big federal, we're doing these big, large-scale, federally funded projects, but the lab's really realizing that we can use a lot of these technologies to answer some really relevant, on-the-ground, applied questions. And so that's where we're headed. And so there's a variety of ways we can do this, either through the laboratory or through UC Berkeley, and there's a Berkeley Lab Foundation. It's not always easy to set up the relationships, but we're successfully doing it with agencies around here, and this is increasing as we hopefully answer and then ask more questions. And so these are some, thank you very much. And so these are some people who are key contacts if you're interested in learning more. That's me. This is Gary Anderson, who's the head of our Ecology Department and molecular microbiology at the lab. And this is Musahid Ahmed, who does all the chemical work. Thank you.
I'm glad you were able to make it. So, you know, this guy totally classes the joint up. So already we've got folks with questions here, and then next there, yeah.
So I'm wondering, have you studied so much the plants that you grow in this compost? Are they absorbing some of the medicine that you're finding in the compost?
Repeat the question if you would.
Yeah, so the question is whether we've actually looked at the plants that are ultimately going to grow in the compost to see if the pharmaceuticals are absorbed in the plants. And we have not done that work. And in fact, where we'd like to go with it is to, you know, take it to the next step. And so once we know what's in the compost and it's going onto the land, what happens in the soil, and ultimately how much of that gets up into the plants. There has been some other research looking at what happened, how much gets into plant matter. But of course, you're talking about there's a ton of different pharmaceuticals out there, so there's a lot we don't know about that. But no, we're not doing that right now.